What if the most powerful cleaning system in human history isn't a drug, isn't a surgery, and isn't even a supplement? It's already inside you, waiting to be switched on. Right now, as you watch this, your cells are accumulating damaged proteins, broken components, and molecular debris that builds up year after year. And most people will never know there's a biological mechanism capable of clearing all of that out. Today, you're going to find out exactly how it works, what the science actually says, and what happens to your body when autophagy runs at full capacity. Every single second, your cells are working. They're producing proteins, generating energy, responding to signals, repairing DNA. But like any system that runs constantly, things break down. Proteins misfold. Mitochondria, the structures responsible for producing your cellular energy, get damaged and start functioning poorly. Toxic byproducts accumulate inside the cell. And unlike the garbage you take out every week, this internal debris doesn't disappear on its own under normal circumstances. Over time, this accumulation becomes a real problem. Dysfunctional proteins clump together and interfere with normal cellular operations. Damaged mitochondria begin leaking reactive oxygen species, which accelerates the very damage they were supposed to prevent. Researchers have linked this kind of intracellular buildup directly to accelerated aging, chronic inflammation, and the early stages of several degenerative diseases, including some of the most devastating conditions affecting people today. Here's what most people don't realize. The body actually has a sophisticated system designed specifically to deal with this problem. It's not something scientists invented in a lab. It evolved over hundreds of millions of years. It exists in nearly every cell of your body. And for most of human history, people activated it regularly without even trying, simply because they didn't have food available around the clock. The question is, are you activating it? In 2016, Japanese cell biologist Yoshinori Ohsumi received the Nobel Prize in Physiology or Medicine for something he had been working on since the early 1990s, a process called autophagy. The word comes from Greek. Auto means self, phagy means eating. So autophagy, quite literally, is the cell eating itself. But that description, while accurate, doesn't capture how extraordinary this process actually is. What Ohsumi and his colleagues discovered is that cells have a highly organized, genetically regulated system for identifying damaged or unnecessary components, wrapping them in a specialized membrane structure called an autophagosome, and delivering them to the lysosome, the cell's recycling center, where they're broken down and their raw materials are reused. This isn't random destruction. It's precise, selective, and incredibly efficient. The cell doesn't just consume itself blindly. It targets what needs to go and preserves what needs to stay. Before Ohsumi's work was fully understood, scientists knew autophagy existed but thought of it mainly as a survival mechanism, something cells did when starving just to stay alive. What the research revealed was far more profound. Autophagy is a continuous quality control process, essential for cellular health even under normal conditions. And when it's working properly, it plays a central role in preventing the kind of cellular deterioration that drives aging and disease. That Nobel Prize wasn't just recognition of a curiosity. It was recognition that this process sits at the heart of how life maintains itself. Your body operates on signals, and one of the most powerful signals that determines whether autophagy is active or suppressed is the presence or absence of nutrients, particularly glucose and amino acids. When you eat, especially carbohydrates and protein, your body releases insulin and activates a molecular pathway called mTOR, mechanistic target of rapamycin. mTOR is essentially a growth signal. When it's active, it tells cells to build, grow, and store. And one of the things mTOR directly suppresses is autophagy. This is where fasting becomes relevant. When you stop eating, insulin drops, mTOR activity decreases. And as those signals quiet down, another pathway, AMPK, which functions as a cellular energy sensor, begins to rise. AMPK acts like an alarm that says, resources are low, we need to be efficient. One of its primary responses is to activate autophagy. The cell shifts from growth mode to maintenance mode. Instead of building new structures, it starts cleaning up the old, damaged ones. This isn't a theory. It's been documented in human and animal studies, published in journals including Nature, Cell, and the New England Journal of Medicine. What makes this particularly interesting is that this switch doesn't require extreme starvation. The body is sensitive enough to nutrient absence that even a moderate fast can begin to shift the balance. The degree of autophagy activation, however, depends on how long the fast continues, the individual's metabolic state, and several other factors, which brings us to one of the most common questions people ask when they first learn about this. This is the question everyone asks, and the honest answer is more nuanced than most YouTube videos will tell you. There's no single number, no magic hour where a switch flips and autophagy suddenly begins. What the research shows is a gradual process. In human studies, measurable increases in autophagy markers have been detected as early as 12 to 16 hours into a fast. But detectable and significant are very different things. Studies using muscle biopsies and blood markers, including research published in Cell Metabolism, suggest that autophagy activity increases substantially between 24 and 48 hours of fasting in humans. Animal models, which allow for more invasive measurement, consistently show peak autophagy activity in the 24 to 72-hour range. A landmark 2019 study from the Salk Institute found that fasting for 24 hours produced dramatic increases in autophagy in intestinal stem cells specifically, cells that are critical for gut health and regeneration. The longer the fast, within physiological limits, the more robust the activation tends to be. What complicates this further is individual variation. Someone who exercises regularly, sleeps well, and has a lower baseline insulin level will likely activate autophagy faster than someone with insulin resistance or metabolic dysfunction. Age also plays a role. Autophagy efficiency naturally declines as we get older, which is one of the reasons researchers believe it's connected to the aging process itself. So while 16 hours is a reasonable starting point, the deeper cellular cleaning the science describes most compellingly tends to happen further into a prolonged fast. Picture this. Inside a single one of your cells, and you have roughly 37 trillion of them, a membrane begins to form around a cluster of damaged proteins. This membrane curves and seals, creating a double-walled vesicle called an autophagosome. It looks under an electron microscope like a bubble wrapping around cellular debris. That autophagosome then travels through the cell and fuses with a lysosome, a compartment filled with powerful enzymes. The contents are broken down into their basic building blocks, amino acids, fatty acids, nucleotides, and those components are released back into the cell to be reused. This process is happening in multiple locations simultaneously within a single cell, and across trillions of cells throughout your body. During a prolonged fast, the rate at which autophagosomes form increases dramatically. Researchers have observed that in fasted states, cells can process significantly more material per unit of time compared to the fed state. This isn't just cleanup for the sake of cleanliness. The amino acids recovered from degraded proteins are used to maintain critical functions, synthesize new proteins, and support tissues that the body prioritizes during food scarcity, including the brain and heart. There's a specific subtype of autophagy worth knowing about, mitophagy. This is the selective removal of damaged mitochondria. And it's particularly significant because mitochondrial dysfunction is implicated in nearly every major age-related disease. When mitophagy is working efficiently, dysfunctional mitochondria are cleared before they can leak reactive oxygen species and trigger inflammatory cascades. Healthy mitochondria reproduce to replace them. The net result is a cell with a cleaner, more efficient energy production system. This is one of the reasons researchers studying longevity have become so focused on autophagy. It's not just about removing garbage, it's about replacing it with something better. The list of what autophagy targets is more specific than most people realize, and understanding it helps explain why this process is so medically significant. One primary target is misfolded proteins, proteins that failed to fold into their correct three-dimensional structure during or after synthesis. Misfolded proteins are not just non-functional, they're actively dangerous. They tend to aggregate, forming toxic clumps that disrupt cellular operations. These aggregates are a defining feature of Alzheimer's disease. The amyloid plaques and tau tangles that accumulate in the brain are, in part, the result of failed protein clearance. Beyond misfolded proteins, autophagy targets damaged organelles, not just mitochondria, but also the endoplasmic reticulum when it's under stress, paroxysms that have become dysfunctional, and even intracellular pathogens like bacteria that have managed to invade the cell. This last function, called xenophagy, is one of the reasons autophagy is considered part of the innate immune system. The cell can recognize an invading bacterium and package it for destruction using the same machinery it uses to clear its own damaged components. There's also a category called agrophagy, the specific clearance of protein aggregates that have already formed. This is particularly relevant to neurodegenerative disease research because the brain has limited ability to replace lost neurons, making clearance mechanisms especially critical. A 2017 study published in Nature Communications demonstrated that enhancing autophagy in mouse models of Parkinson's disease significantly reduced the accumulation of alpha-synuclein, the protein that forms the toxic Lewy bodies characteristic of that condition. The implication is significant. Autophagy isn't just passive maintenance. When functioning at a high level, it actively works against some of the most feared diseases in modern medicine. The relationship between autophagy and cancer is one of the most actively studied areas in current biomedical research, and also one of the most complex. Early findings showed that autophagy genes, particularly BECLIN-1, appear to function as tumor suppressors in certain contexts. Mice with reduced BECLIN-1 expression develop tumors at significantly higher rates than control groups. The underlying mechanism researchers propose is straightforward. When cells lose the ability to clear damaged components efficiently, DNA errors accumulate, cellular stress increases, and the biological conditions associated with malignant transformation become more favorable. A 2011 review in Nature Reviews Cancer identified autophagy deficiency as a recurring feature across multiple cancer types in studied models. In Alzheimer's research, the findings are equally significant. Work from the Nixon Lab at NYU, among the most referenced groups in this field, documented that autophagy activity is severely impaired in the neurons of Alzheimer's patients. Autophagosums were observed accumulating inside neurons without completing the degradation process, resulting in a buildup of cellular material rather than its clearance. Subsequent studies in laboratory models found that interventions aimed at restoring autophagy function were associated with reduced amyloid accumulation and changes in cognitive markers under controlled experimental conditions. These findings have positioned autophagy as a subject of active investigation in several drug development programs, currently in clinical trials. On the aging front, the data from multiple independent research groups points in a consistent direction. In one widely cited experiment, genetically enhancing autophagy capacity in mice was associated with an extension of healthy lifespan in the range of 10 to 15%, without any additional intervention. Research conducted in C. elegans, a model organism used extensively in longevity studies, found that when autophagy genes were blocked, the lifespan extension typically produced by caloric restriction was completely eliminated. That finding carries a specific implication. The longevity effects associated with eating less may operate, at least in part, through autophagy as an intermediary mechanism. Across multiple model organisms, the data suggest autophagy isn't merely correlated with healthy aging. It may be a required component of it. The single most effective way to suppress autophagy is to eat frequently. Every time you consume food, particularly anything that raises insulin or activates mTOR, you send a signal that tells the cell to stop cleaning and start building. This is entirely appropriate in the context of a meal. The problem is that modern eating patterns, characterized by 3 meals plus multiple snacks, spread across 14 to 16 waking hours, mean that mTOR is chronically elevated and autophagy is chronically suppressed. The cellular cleaning system that took hundreds of millions of years to evolve barely gets a window to operate. Specific nutrients are particularly potent autophagy suppressors. Leucine, an amino acid abundant in dairy, meat, and protein supplements, is one of the strongest direct activators of mTOR known. This doesn't mean protein is bad. It means that consuming protein, especially in large amounts, will reliably shut down autophagy. Glucose has a similar effect through insulin signaling. This is why some researchers studying autophagy are skeptical of approaches like fasting with protein shakes or modified fasting protocols that include significant caloric intake. The molecular signals those nutrients send may substantially blunt the autophagy response even if total calories are reduced. Chronic sleep deprivation is another underappreciated factor. A 2019 study published in Nature Communications found that sleep loss impairs autophagy in the brain specifically. An alarming finding given that neuronal autophagy is critical for clearing the metabolic waste products that accumulate during waking hours. The glymphatic system, the brain's own waste clearance mechanism, operates primarily during deep sleep, and it works in coordination with autophagy at the cellular level. Stress, through chronically elevated cortisol, also suppresses autophagy pathways. The irony is that the lifestyle factors most associated with poor health, poor sleep, chronic stress, constant eating, are the same ones that keep the body's most powerful internal cleaning system permanently offline. The most evidence-backed approach to activating autophagy remains time-restricted eating and periodic prolonged fasting. For baseline autophagy stimulation, compressing your eating window to 8 to 10 hours per day, effectively a 14 to 16-hour fast each night, appears to be sufficient to produce measurable effects over time. For deeper activation, 24 to 72-hour fasts conducted periodically, monthly or quarterly, depending on individual health status, are what the research most consistently associates with significant autophagy upregulation in humans. Exercise is one of the most underrated autophagy activators. A landmark 2012 study published in Nature by Beth Levine's group at UT Southwestern demonstrated that exercise induces autophagy in multiple tissues simultaneously, including muscle, liver, and brain. The mechanism involves both AMPK activation and mechanical stress on muscle fibers, which triggers selective autophagy of damaged contractile proteins. Interestingly, the combination of exercise performed in a fasted state appears to produce an additive effect, activating autophagy through 2 independent pathways at the same time. This is one reason why morning exercise before breaking a fast has gained attention among researchers studying longevity. Certain compounds have also shown promise in autophagy research. Spermidine, found naturally in wheat germ, aged cheese, and mushrooms, has been shown in multiple studies to induce autophagy through a pathway independent of mTOR inhibition. A 2018 clinical study published in Cell Reports found that higher dietary spermidine intake was associated with reduced mortality and better cognitive function in older adults. Resveratrol, found in red grapes and berries, activates SIRT1, a deacetylase that promotes autophagy gene expression. And metformin, a diabetes drug now widely studied for longevity, activates AMPK and has demonstrated autophagy-inducing effects in multiple tissue types. None of these are substitutes for fasting, but the research suggests they may work synergistically with it. What follows a significant period of autophagy activation is, in many ways, as interesting as the process itself. When fasting ends and nutrients become available again, mTOR reactivates, but now it's operating in a cleaner cellular environment. The raw materials recovered during autophagy become available for the synthesis of new functional proteins. Mitochondrial biogenesis, the production of new mitochondria, increases. Cells that have cleared their damaged components are essentially refreshed, operating more efficiently than they were before the fast began. This refeeding phase is not just a return to baseline. Research in stem cell biology has shown that prolonged fasting followed by refeeding triggers stem cell activation in multiple tissues. A 2014 study from Valter Longo's lab at USC, published in Cell Stem Cell, found that cycles of prolonged fasting caused immune system stem cells to regenerate, effectively producing a partial renewal of the immune system. Similar effects have been observed in intestinal stem cells. The implication is that the fast refeed cycle isn't merely a cleaning event, it's a regeneration cycle where old and damaged components are removed and new, functional ones take their place. The long-term picture painted by the research is one where periodic fasting, and the autophagy it activates, functions as a kind of biological reset. Not a cure, not a guarantee, but a fundamental maintenance process that the human body evolved to perform and that modern life has largely disrupted. The science doesn't promise immortality. What it does suggest consistently and across multiple research groups and model organisms is that keeping this system active is one of the most impactful things a person can do for their long-term cellular health. And unlike most interventions in medicine, it costs nothing. It just requires knowing it exists and choosing to use it.